Hello everyone, and a warm welcome to this webinar on the topic Tailoring Immunity, Defining the Impact of Vaccine Type and Route of Administration. This webinar is part of a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. My name is Clive Nyaribo, Assistant Research Officer at KEMRI-Wellcome Trust Research Program, providing support to the Vaccine Workforce Development Program. Together with my colleagues, Professor George Warime, Marianne Munena, and Ravenda Nafura, welcome you to this session. Before we get started, a few housekeeping matters. Kindly take note that this webinar is being recorded. Kindly feel free to use the chat option to share your questions during the presentation, and we will address them during the Q&A session. We'll have a few open slots for people to directly ask questions to the presenters. If your internet is stable, you can raise your hand using the available icon. As Arya mentioned, this is a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. This being run under the KEMRI-Wellcome Trust Research Program, which is a world-renowned health research unit of excellence based in Kilifi and Nairobi in Kenya, as well as Mbare in Uganda. Over the years, we have excelled in the use of novel ideas and collaborations to achieve better health for Africa, while also developing African scientific leaders. Our vision at the program is to undertake cutting-edge and novel research relevant to national, regional, and global needs. It's under this program that we have the Vaccinology Workforce Development Program that aims to expand the technical know-how and capability to sustainable end-to-end vaccine research and development in Africa through training and knowledge exchange activities in the vaccine ecosystem. This involves areas such as target product profiling, research and discovery, preclinical and clinical studies, biomanufacturing, regulatory affairs, and deployment. The workforce development is through three channels. One, we have internships at vaccine manufacturing institutions that aims at establishing a mentorship scheme that links interns with senior vaccinologists and healthy systems and engagement with academics globally to share and strengthen experiences in vaccine research and development and manufacture and enhance career development. Two, we have a full funded master's level course in vaccinology where applicants are selected based on their direct involvement in vaccine research and their professional excellence and academic achievements. And three, we have webinars like this one, where a career scientists, graduate and undergraduate university students in Africa to provide information on possible career options for university students for informed career decision making. This program is in alignment with the Africa Union's platform for harmonized African health products manufacturing, ambition to increase local vaccine production to meet 60% of the Africa's routine immunization needs by 2040, and to strengthen the capability for vaccine research, discovery, and manufacture in Africa. And this webinar series is part of this engagement. Today, we are glad to have Alexandra Spencer, who will be discussing the topic Tailoring Immunity, Defining the Impact of Vaccine Type and Route of Administration. Dr. Spencer is a senior lecturer at the University of Newcastle and leads the Vaccination and Infection Immunology Research Group based at the Hunter Medical Research Institute. Following a PhD at the Centenary Institute in 2006, she moved to the University of Oxford to apply her knowledge of T cells to vaccine development. In her time abroad, Dr. Spencer was involved in the development of new adjuvants, vaccine vectors, and vaccination regimens across a range of vaccine programs, including malaria, influenza, and SARS-CoV-2. The translation focus of the Jenner Institute enabled her to follow these approaches from preclinical studies through to clinical trials, working with academic and industry partners. In 2022, Dr. Spencer moved back to Australia to establish a research group at the University of Newcastle, where our group is focused on understanding how early changes in immune activation, driven by the choice of vaccine or route of administration, shapes the quality of adaptive immune responses. Over to you, Alex.
So thank you very much for the introduction. Let me share my screen. Can you see that all properly?
Yes, we can.
Perfect. So thank you very much for the invitation to present today. So yes, I now work at the University of Newcastle, but I am a university lecturer and also a researcher, so I split my time between two places. My research group is located here at the Hunter Medical Research Institute, and when I'm teaching, I am teaching over on campus here at the Callaghan campus. So Newcastle is about 2 hours north of Sydney on the east coast of Australia. It's nice and along the coastline. It's really lovely here because after living in the most inland city in the UK for 16 years, I'm back on the coast, which makes me very, very happy. So today I'm going to talk about a lot of the work that I did when I was based in Oxford, and then also introduce some of the work that we've been doing since I came back to Australia 3 years ago, and from some researchers in my group. I'm going to try and highlight along the way some of the people who've really been involved in this work. Before I start, I do just want to acknowledge the traditional custodians of the land. So the University of Newcastle actually sits on a pretty large geographic footprint in New South Wales and Australia, which means we sit on the lands, the traditional lands of the Awabakal Nation, the Dikindjin Nation, the Biripi Nation, the Wirimai Nation, Woonora Nation, and the Eora Nation. And so I just would like to pay my respect to the wisdom of elders past, present, and emerging. And I acknowledge that the Pambalong clan of the Awabakal Nation and the Wirimai people were the first scientists on the lands where I work and live. Okay, so we don't need a global pandemic to, well, a global pandemic really highlighted how much of an issue infectious diseases are. But even before the COVID outbreak, the WHO have already estimated that 23% of deaths that we see worldwide annually are really due to infectious diseases. I mean, obviously the deaths really relate to where you're which country you're living in. But if we take all deaths in general, we see that it's over 20% due to infectious diseases. And I think what's really upsetting is that when we look at children under the age of 6, we actually see that 64% of these are due to infectious diseases. But what we know that vaccination has really made a really big global impact in terms of reducing the morbidity and mortality in terms of infectious diseases. And in fact, we've actually been able to globally eradicate polio through the implementation of vaccination programs. So I really like this photograph. I often start my talks with it because I think it's a really clear visual demonstration of the impact of vaccines. So this photograph was taken in 1901 by I've forgotten where this surgeon was based, but he took this photograph because he had there were 2 boys present at the time. Both have been exposed to smallpox. But what you can see here is that while one boy had really succumbed to a smallpox infection and he has all these pox scars all over him, the boy sitting right next to him, although he was exposed to smallpox at the same time, seems to not really have any lesions on him at all. And this is really due to the fact that the boy here on the right had been vaccinated against smallpox in infancy, and that smallpox vaccination had protected him from being infected and succumbing to infection with the smallpox. So really, really clear visual demonstration about the impact of vaccines. When we think about vaccines for infectious diseases, effectively the process that we're doing has been around for a really, really long time. And initially, it wasn't called vaccines. It was actually a process called variolation. The process of variolation was occurring in the 1700s and earlier. And what it was is that if you took a very, very small sample of smallpox from one of those pustules, and you scratched it onto the skin of someone who was naive or hadn't been exposed to, it was known that if you got the amount of smallpox pustule correct, it didn't induce that person to become really, really sick, that individual would be potentially protected from smallpox. And it was a process that was occurring in China, in India, in sub-Saharan Africa, in Turkey. And actually there's some really good evidence of people taking this variolation process and passing it on to what we would today classify as high-income countries, and then how they've used that to prevent smallpox. So the really lovely example I like to give is Wanzamus, who happened to be an African slave who got taken to Boston, and he passed on this technique to his owner, and it's really been reported as making a huge impact in terms of a smallpox outbreak that occurred in 1721. And when we think about vaccines today, we often go back to Edward Jenner. because what he did kind of termed the coin vaccine. And so Edward Jenner in 1796 was a general practitioner, but he was also a farmer, and he was based out in Gloucestershire, which is about an hour and a half of Oxford in the UK. He lived in the beautiful countryside, and he ran cattle. And what he noticed is that he ran these cattle, and he obviously had milkmaids who would milk his cows for him. He noticed that his milkmaids had the scars of which were very, very similar to the smallpox scars. He also knew that his cows often displayed very similar symptoms, so these pustules that were also seen in smallpox. So he kind of realized that actually cows also got a version of the smallpox, but it was specific to cows. And perhaps it was these milkmaids that they were being exposed to cowpox that seemed to protect them from smallpox. Because I forgot to mention, although all the milkmaids also had these cowpox scars, known that these milkmaids never seemed to become infected when there was a smallpox outbreak. So Edward Jenner tested his theory by taking some smallpox from one of his cows, and he administered it to a child who happened to be the son of one of his farm workers. And he was later able to demonstrate that when he exposed James Phipps to smallpox, it protected him from smallpox. And it was the first documentation of the process called vaccination, where you could administer something and it could lead to protection against disease. And that's how we get the actual term vaccine. It's taken from the Latin word of cow called vacca. So obviously that was one of the first clinical trials, and we definitely wouldn't be doing that this day. Now we actually think about, we sit back and think about design of vaccines in a very different way. And so when we're developing a vaccine, some of the features we're really trying to look for is, first of all, we need to make sure that that vaccine is safe. There's no point having a vaccine to protect people against an infectious disease when administering the vaccine actually causes more damage and more harm than good. What that vaccine also needs to do is it needs to be protective from the infectious disease. And the way that it's protective is it's able to induce an effective immune response. And now that type of immune response that it needs to be protective is actually going to depend on the pathogen that you're trying to make a vaccine against. So I call this as being pathogen dependent. So for instance, if you're developing a vaccine against an extracellular pathogen, so one that sits outside of the cells, say for instance a bacterial or extracellular parasite, say for instance a protozoa or a fungi, then perhaps what you really want to do is you want to induce an antibody-mediated response. So you want to induce high levels of antibodies that can block and neutralize that extracellular pathogen. In contrast, if you're developing a vaccine against an intracellular pathogen, so one that's able to infect cells and reside within the cells, then perhaps what you're really trying to focus your vaccine to do is to make a vaccine that induces cell-mediated immunity. So that's primarily T cells that can locate those infected cells and kill them. So again, depending on the particular pathogen that you're making a vaccine against, you really need to make sure that it's inducing the right effective immune response. But there's also some very important practical considerations that we need to take into account. We have to think about the cost of manufacturing and the cost of the final product. Again, because there's no good making a vaccine that's too expensive that then no healthcare budget can accommodate that vaccine. It also needs to be stable. There's no point having a vaccine that will go off within a day, because then you're never going to be able to get it out to where it needs to be. It's important to consider how you deliver that vaccine. So, obviously giving it through a readily accessible site is going to be better than something that, really requires a specific technique. And again, we really need to consider the side effects. It goes back to the safety of the vaccine. We need to think about what is the impact of the vaccination process. And the reason I put this up here is because I've already highlighted the fact that the type of pathogen that you're dealing with is going to depend, therefore going to make a decision about the type of immune response you want to make. But also we're starting to think that We also have to really think about the target population, because a vaccine you're making for children under the age of 5 might have very, very different considerations in terms of its side effects compared to, for instance, a vaccine that you're making against prostate cancer for men in Australia. You might be thinking about very different side effects or practical considerations in that context. And what I should also say is what we're starting to really think about now is the site of infection. So if we're trying to make an immune response that's really targeted to the mucosal site, then perhaps we might be looking for an alternative delivery route, and we might be considering different side effects. So the traditional types of vaccines that we've seen on the market, those include things like whole inactivated vaccines, and that's arise through that traditional sense where the virus, the bacteria, or the other pathogen has been grown up in culture. And then it's effectively being killed either through heat, chemical, or radiation. Some good examples of that is some types of polio vaccines, some types of rabies vaccines, and some types of hepatitis A vaccines. We also have our live attenuated vaccines. And again, traditionally, they have arisen because the virus, the bacteria, or the pathogen has been grown up in culture. And it's been weakened in some way. So it's still alive and it can infect, but it's weakened so that you don't get the pathogenicity that you see. Now, sometimes that has occurred because of the constant passaging of that pathogens through a cell line has just meant that it's lost a lot of its virulence. Or we might do that through genetically attenuating that particular pathogen by deleting out some of its genes. And so some of the examples of those vaccines that we have on the market, again, is some types of polio vaccines, MMR, chickenpox, the yellow fever vaccine, the rotavirus vaccine, some versions of influenza vaccine, and some versions of tuberculosis. We also have our protein subunit vaccines. And again, traditionally, the way they've come about is the virus, the bacteria, or the parasites being grown up in culture, and then it's purified a single protein out of that cell supernatant. That would be the traditional, the way that we've developed these protein-based subunits. Or these days, what we might do is we might insert that gene into a cell lines, and then we would generate the recombinant protein. And again, we have some vaccines on the market. These include hepatitis B, diphtheria, meningococcal, and influenza. And finally, what we've seen more in the last couple of years is this particular type of vaccine, which is called a virus-like particle. So it looks like a virus, but it's not quite a virus. So it has the structure of a virus, but it doesn't have the genome. So it still stimulates the immune response like it's a virus, but it can't actually replicate. And the two licensed vaccines that I'm going to say this is prior to 2020, the licensed vaccine that we have is the human papillomavirus and the hepatitis B vaccine. And now these vaccines have really kind of been generated from these culture passaging and making things better and cleaner along the way. And they've worked really well because they've traditionally worked through the production of antibodies. They're highly effective in inducing antibodies, which are able to neutralize and really protect against those extracellular pathogens. But what about those making a vaccine where you have one of those diseases that requires cell-mediated immunity? And that's because potentially you're dealing with an intracellular pathogen. And so one very good example of that is if you're developing a vaccine against malaria, but really quite specifically if you're making a vaccine against the liver stage of malaria, which is where the sporozoite has infected the hepatocyte and is undergoing asexual replication. And here again, you might think about where you want to target that immunity. So if you want to make antibodies against the sporozoite to start with, you might be thinking that the site that you need to get to is the skin. Or if you're trying to make cell-mediated immunity in the liver, you might be really trying to target that to the liver where those parasites going asexual replication. But it's not just liver stage malaria. There's lots of other diseases that we think protection is really mediated by cell-mediated immunity. Another good example is tuberculosis. So it's a bacterial infection, but what happens is there's this granuloma that forms and kind of it protects that bacteria from T cells and antibodies. And I highlight this here just to point that, that's occurring in the respiratory tract. What about HIV? So this is where you've got infection of immune cells, such as the CD4 T cells. You know, once the virus has infected the cells, the antibodies aren't going to mediate protection. So perhaps you're trying to make cell-mediated immune response. And then I also put up an example of influenza. So current vaccines against influenza are all about inducing antibodies to neutralize it, but we actually know that T cells are really important in the context of protection against influenza. So perhaps what we really want to do is we want to make a vaccine that can induce both cell-mediated immunity and humoral immunity. So what type of vaccine platforms can you use for those? Okay, so I'm going to switch back to malaria because I spent a long time looking at how we develop malaria vaccines. So as you will all be aware, in humans, the way we get infected is through the bite of an infected mosquito, and that infected mosquito deposits sporozoites into the bloodstream that they make their way to the liver, where they undergo asexual replication before they convert into a blood stage form and they have the merozoites. And it's when those, where we see the symptoms of malaria is through that blood stage infection, whether you have the cyclical infection and then release of merozoites. At a certain point in the blood stage infection, you start to develop the parasite starts to actually develop sexual, so it's gametocytes. And if another mosquito comes along and takes it up, that'll go into the midgut. That there goes sexual replication, and then you have the cycle going back. So those oocysts then lead to sporozoite production. The sporozoites go back into the salivary glands and so forth. And the complexity with malaria is that not only does it have two hosts, so it has its mosquito host and it has a human host, it also has lots of different life cycles. So you have the sporozoites for the pre-erythrocytic stage, that they express a very, very different set of proteins to what is expressed during the blood stage. So, and this also means that where the parasite is very different, the proteins are very, very different, but also the type of immune response that you need to protect against the pathogen at those different stages can also be very different. Okay, so if we were trying to block the sporozoite when it's just been released from the mosquitoes, what's known that actually inducing antibodies can actually lead to protection, but you need excessively high levels of antibodies because you have to block that sporozoite in the short time between being bitten by a mosquito and those sporozoites making it to the liver. If it's a blood stage infection, again, antibodies can protect and neutralize the merozoite, but again, high levels of antibodies are probably required so that you can catch those merozoites in the short time between their release from a red blood cell and they go on to infect another red blood cell. And there's also programs looking at development of what we call transmission blocking vaccines. And here, if you can induce antibodies against the sexual stages in a vaccinated individual, then when they're bitten by a mosquito, those antibodies get taken up with the blood meal. And then the development of the parasites in the mosquito is actually halted. So these are all antibody mediated, we think, so they could, use the same particular type of vaccine platform to induce those. But if we're talking about making a vaccine against the liver stage of malaria, then really what we were focused on, it was trying to induce cell-mediated immunity, in particular CD8 affected T cells, so that they can locate and destroy those infected hepatocytes. So why not just make a whole parasite vaccine? They've been really, really highly effective for lots of different diseases. Well, actually, one of the best malaria vaccines that was that's been shown in human clinical trials to date was actually some original work done in the 60s and 70s, where sporozoites that had been weakened through irradiation were able to induce really, really strong protective immune responses. So Nussensweg demonstrated in mice that if you irradiated sporozoites and gave them to mice, it could induce significant immune response that could protect when these mice were then given another round of malaria parasites. And then Clyde showed that this was highly effective in humans, that if you gave human irradiated sporozoites, that you could protect them. And it was known that this administration of irradiated sporozoites could lead to high levels, could lead to protective T cells and also antibodies. But in terms of deployment, maybe it's a highly protective vaccine, but maybe it's not the most practical vaccine. because one of the problems is that although their sporozoites are irradiated, they really still need to be infectious to induce that immune response. And so it requires isolation and purification of those mosquitoes for the salivary glands. It really requires cryopreservation, so putting those sporozoites into a protective solution and then freezing them down to minus 170 degrees. It means that deployment is more difficult because you have to take a tank around with you. And also what's been seen with the human clinical trials is that you need much, much higher dose. So it's not going to be a really global rollout vaccine because it's just got a few too many challenges along the way. So what about, then we look at vectored vaccines. So what a vectored vaccine is, we take an individual protein for that particular pathogen and you put it in a vector. And then when you vaccinate with the vector, it triggers the production of the protein through normal cellular pathways. And then when that protein gets made, because it's a foreign protein, it can lead to the induction of an immune response against that introduced protein. And so there are vaccines on the market which would classify into these vectored vaccine categories. If we think about COVID vaccines, we have Pfizer and Moderna mRNA-based vaccines where these lipid nanoparticles deliver messenger RNA that leads to the production of the protein and they have mounting of immune response. We have the Oxford AstraZeneca vaccine. We also have the Johnson & Johnson vaccine, which is a virus-based vaccine where the spike protein has been encoded within the viral vector. And once administered, you get production of that spike protein immune response. And the same with the DNA. What I forgot to mention is that, the key point about the vectored vaccines is they already retain a lot of those immunostimulatory components, such as it's the viral particles that triggers the innate immune response. And as a consequence of production of the protein plus stimulation of the immune response, this innate immune response, you get an adaptive immune response against that introduced protein. And so in Oxford, we were really focused on using vectored vaccines, in particular viral vectored vaccines, because we knew that they were really effective at inducing cell-mediated immunity. So in my time in Oxford, we tried many, many things. And also the work had started long before I got to Oxford. So we've tried lots of different viruses as the vector. These include adenovirus vectors, modified Vaccinia Ankara, which is a pox virus. It's a DNA-based vector. I've also tried adeno-associated viruses. It led to the generation of ChAdOx1, so a chimpanzee-based adenovirus that we then made into a GMP stock. This was the backbone of what was the Oxford AstraZeneca COVID vaccine. It's called ChAdOx1. We also made another chimpanzee adenovirus called ChAdOx2, which is the basis of the Oxford rabies vaccine, and I've now forgotten which other ones that's gone into. But we also tried other types of vectors. We generated virus-like particles, R21, and in fact, it's the R21 that has become the Oxford malaria vaccine, this virus-like particle expressing a CSP protein from P. falciparum together with an adjuvant. Some of the other strategies we tried to induce or enhance immune responses was co-encoding adjuvants together with the antigen that we were interested in. We've also did lots of screening programs to identify new antigens or look for combinations of antigens. And we also looked at ways that we could mix up our viral vectors to induce the largest immune response. We changed the interval. Because we were really focused on inducing CD8 T cell responses in the liver, we also looked at ways that we could target immune responses to the specific sites. So what did we learn along the way through testing all of these different strategies? Well, the first thing I'm going to tell you is the type of immune response that we saw was really, really dependent on the particular vaccine that we used. So this is an example of where we have the same pathogen protein expressed in 4 different types of vaccines. So it's the CSP protein from P. falciparum. So here we have 2 viral vectors that we know if we vaccinate with an adenovirus, and we come along for at least 4 weeks later with a pox virus, that we know that we can get really good T cells and we can also get antibodies. This is the R21-based vaccine, so it's a virus-like particle that we deliver together with an adjuvant, and if you give 2 doses of that, it can be highly protective. But we also compared it to some irradiated sporozoites, that traditional whole parasite vaccine, that in this case, it was a transgenic parasite that expressed the CS protein. And so now if you look at the humoral immune response induced by this, you can see adeno followed by MVA gives us not a bad antibody response. That's pretty good for a viral vector. If we use a virus-like protein in an adjuvant, we see the highest level of CSP-specific antibodies. And if we use those irradiated sporozoites, we get a pretty good immune response relative, you know, that's considering we're vaccinating with a whole parasite. We get a similar level of antibodies as we're seen induced through 2 viral vectors. So that's the antibody response. We're already seeing differences depending on the type of vaccine that we use. And when we start to look at the cell-mediated immune response, so looking at CD4 helper T cells or cytotoxic CD8 T cells, we see this really big difference in immune responses depending on the vaccine. So none of these particular ones are great in inducing CD4 T cells. We see some with our AD-MVA. We see slightly less with our R21-based vaccine, and we see almost not very many CD4 T cells with irradiated sporozoites. If we look at our CD8 effector T cells, we see super high response, antigen-specific response with our viral vector combination compared to R21 and irradiated sporozoites. So with these different types of vaccines, we're seeing a really different pattern in their adaptive immune response, which is dependent on their vaccine. Okay, so what's the summary of that? Well, if you want to induce antibodies using a virus-like particle together with an adjuvant, gives you the highest level of antibodies. If you really focused on T cells, then using the viral vectors gives you much better T cells, can also give you some antibodies. But the irradiated sporozoites can give you lots of antibodies, maybe not so many T cells when we're just looking at one particular antigen. Okay, this is some other work that that one of my recent honours students has done, and this is really looking at the types of antibody responses that are induced when you give different types of adjuvants. So here in this particular experiment, I use one type of adjuvants. There's also a whole, apart from there being lots of different types of vaccines, so viral vectors, virus-like particles, or radiated sporozoites, even within proteins and adjuvants, there's a huge variety in the different types of adjuvants you can get. Anyway, in this particular experiment, what we did is we took the same protein and we administered it to mice with different types of adjuvants. So in this case, it was Adavax. In this case, it was a Quil-A, which is similar to Matrix M. And this one, it was Al-Hydrogel, so very similar to alum. And we also compared it to our viral vectors. And when we looked at the antibody response, so this is looking at the total amount of over-specific antibodies, you can see a little bit of a difference. So you see it's not statistically significant, but our viral vectors induce a slightly lower antibody response than our protein in adjuvants, although maybe not the Quil-A. But importantly, when we look at the function, at the different types of immunoglobulin subclasses, so this is total IgG. If we then start to look at immunoglobulin subclasses, so IgG1, which is we might think of as a type 2 antibody response, or IgG2 or IgG3, we see that when we normalize for the amount of antibodies that are induced in these mice, we see a different pattern of their subclasses within that immune response. So we see that while these protein and adjuvants typically shift the antibody response towards an IgG1 response, our viral vectors induce much more of a broad immunoglobulin subclass response. So we see both IgG1 and IgG2, and no one really makes a lot of IgG3 in this particular strain of mice. So how does changing the immunoglobulin subclasses then impact the functionality? So what we did is we set up ELISAs that can measure the ability of antibodies to induce the complement pathway. And you can see here that when we normalize for the level of total IgG, we can see that when mice are vaccinated with viral vectors, we see a much a higher capacity for these antibodies to bind to complement, so potentially having a different way that they function. The other thing that we did is we also set up a phagocytosis assay, so looking for the ability of these antibodies to induce engulfment, and in this particular case, it's beads coated with antigen, so a functional response, so phagocytosis and engulfment of these. And you can see that when we normalize for antibodies, we see almost equivalent phagocytosis when you have protein in adjuvant versus a viral vectors. But knowing that this is normalized for the total amount, what we actually see is that the viral vectors are changing the functionality. So the summary is that it's not just the level of antibodies that can change with the type of vaccine, it's also the quality and functional capacity of the immune response within that compartment. Okay, so I'm going to harp back to this point that immunity is really driven by the vaccine platform. Here we're looking at T cell responses. So you can see here, this is just a single dose vaccination with MVA. We don't really need to, so this is, we had one MVA compared to an EDVA with an internal adjuvant. The point being, or the point I want you to pick up here is when you look at these immune responses, you can see that a lot of the response in following MVA, you see very, very good CD4 T cell responses that can be up to almost 1% of the circulating CD4 T cells are specific for the particular antigen, whereas the CD8 response in this particular case is less than 1%. Whereas if you vaccinate with an adeno-based virus, which is similar to the backbone of the Oxford AstraZeneca vaccine, here the CD4 T cell responses are a little bit less than the MVA, around 1%, but you can see that you see much, much greater CD8 T cell response. So MVA viral vectors really drive T cell responses towards inducing CD4 T cells, whereas an adenoviral vector really shifts the immune response towards a CD8 T cell response. But importantly, you can actually get the best of both worlds through a combination of these viral vectors. So if you vaccinate mice with an adenovirus and you come back 4 weeks later with a pox virus, actually now what you can do is pretty good CD4 T cell responses, pretty good CD8 T cell responses, and more so what you can induce is pretty good antibody responses. So the combination of two different types of vaccines can actually give you the best of both worlds, so T cells and antibodies. And this was a work of a PhD student in Oxford, Benedict Halbroth. This just touches on the point that it's also that we've also saw this in the context of COVID vaccines. So this was a study where we were comparing our adeno-based viral vector compared to an RNA-based vaccine. And the take-home message is that when you combine the both of them, you can induce antibodies and you can induce T cells. So it's a really nice way to kind of broaden your immune response. So going back to malaria, we tried lots of different ways to induce CD8 T cells. Before I came to Oxford, there was a lot of work using a different type of pox virus, which was cowpox. It gave a few T cells, but really gave no protection against malaria challenge. Worked a lot with they worked a lot with DNA-based vectors. You had to give a triple dose of it, gave a few T cells, but really gave no protection against malaria. And it wasn't until they started working with viral vectors that they started to see increasing T cell responses. And it was when they started, when we started using adenoviruses, so that was about the time that I joined Oxford. Now we started to see decent numbers of T cells, and those decent numbers of T cells also translated into protection. So through the use of adenoviruses and adenoviruses plus MVA, we started to see really, really good T cell responses in volunteers, and this corresponded to protection. But although we were getting protection, it still wasn't high enough. So how could we actually improve these vaccines to generate much better T cell responses and protection against malaria? What we do know from preclinical studies, and kind of we're getting that way with our human studies, is that the more T cells that were able to be induced by vaccination actually led to greater levels of protection. And this is just a study in mice where we can actually take T cells from a vaccinated mouse and we can transfer them to a recipient mouse, and we can really transfer just a very, very specific number of T cells. And what we see in this particular study is that the number of T cells transferred really correlates with protection. So the more antigen-specific T cells there are, the better protection there is. And what we actually looked at by measuring T cell responses in the liver of these mice, it really depended on how many of these T cells made it into the liver during that sporozoite infection. So you can have high numbers of circulating T cells, but those circulating T cells also need to make it into the liver to find those infected cells to clear the infection to really lead to protection. Where does that lead? Well, it led to a project really looking at ways that we can target that CD8 T cell response to be in the liver and reside in the liver long term. It's slightly challenging to do that because the liver is constantly surveying antigens that come from the gastrointestinal tract. And so if you give too high a dose of your vaccine in the liver, it can actually mean that your T cells don't respond properly. Some people had looked at alternative ways to bring T cells into particular sites. There was some work by Shinone Suwaki that actually was able to demonstrate that you could activate affected T cells and then pull those CD8 affected T cells into a mucosal site by delivering UV inactivated chlamydia. So this is in the context of a chlamydia model. It was also seen in the context of trying to drive T cells to a mucosal site, that actually if you induce affected T cells through systemic vaccination, you could actually pull these affected T cells into a particular mucosal site. And so we hypothesized, well, we know that viral vectors are really good in inducing circulating responses. Maybe we can look at strategies where we can pull those circulating T cells into the liver, and that might lead to better protection. And maybe the way that we do that is that we can drive T cells into the liver by delivering antigen into the liver. So I had a really amazing PhD student, Anita Gola, who really drove this particular project. And she looked at ways that we could deliver antigen into the liver, but to get around the problem of tolerization, first you would induce affected T cells through systemic immunity, and then you would drive those T cells into the liver. So to induce that circulating affected T cell response, she would vaccinate with an adenovirus expressing the antigen that we were interested in this case, it's ovalbumin, and through intramuscular vaccination. And then she would try to target those affected T cells into liver by delivering nanoparticles loaded with ovalbumin and given intravenously, because we were able to demonstrate that if you give these nanoparticles intravenously, they get picked up by the cells in the liver and you can get antigen expression in the liver. So when she did that, what she saw is that if you prime that immune response and then you target through intravenous injection, you can dramatically increase the number and frequency of those antigen-specific T cells in the liver. So you can see we can increase the frequency of the antigen-specific T cells in the liver up to about almost 20%, compared to 3% if we just give a single intramuscular vaccination, or compared to 3% if we give an intramuscular vaccination followed by delivery of those nanoparticles intramuscularly. And what she discovered also when we went on and then challenged these mice is that it was only through targeting those T cells to the liver that you actually were able to be protective. And so by targeting these T cells to the liver through antigen delivery, she was able to increase the number of antigen-specific cells, and she was really able to improve protection against a malaria challenge. So by she could shift the immune response to the liver by delivering antigen to a specific site. So although she originally did the work with nanoparticles, we always kind of knew that some of these viral vectors we were using with actually really kind of liked some of the hepatocytes in the liver. So in this particular experiment, she was comparing different viral vectors. So adenovirus compared to a chimpanzee adenovirus compared to our pox virus. And she administered these vaccines intravenously. And in this case, the particular antigen she used meant that we could image how much antigen was being expressed through whole mouse imaging. So vaccines express luciferase, and then we can pick that signal up by taking a photograph of the mice while they're under anesthesia. The point I want to show is that when you give adenovirus intravenously and you give chimpanzee adenovirus intravenously, what you can see is we can see a super, super high level of antigen in the liver that slowly starts to decline with time. Whereas if you give MVA, you can see a little bit of a signal in the liver and then it rapidly declines. And that's what you're seeing over here. So you're seeing a lot of antigen, then a slow decline with both types of adenoviral vectors, whereas you see a dramatic loss in the level of antigen expression when you give MVA. So it meant that because these viral vectors were able to deliver antigen to the liver, that perhaps we could use these as our targeting vectors as well. So what Anita went on to do is she would prime mice with an intramuscular vaccination, and then she would target T cells into the liver through an intravenous injection of adeno or MVA. And when she did that, again, like what we saw with the nanoparticles, you could dramatically increase the frequency and total number of antigen-specific T cells in the liver. And it also led to much, much higher levels of protection. So 3 weeks post-vaccination and 2 months post-vaccination, highly effective. And we went on to demonstrate that it was actually a very particular population of liver resident memory cells that could mediate this protection. So we started to look at strategies that would really specifically induce these populations. Again, going back to the concept of you've got to think about the features that you need for an effective vaccine. Intravenous administration is possible, but it's really not something that you'd want to use as a global vaccination program. It is being used in some particular programs. So Scenario, who make the irradiated sporozoite vaccine, demonstrated in trials that you could do direct venous administration. It does require highly trained staff, but it actually was quite well tolerated. We also know that if you inject too high a dose of adenovirus intravenously, it can be really toxic. This was actually done in some gene studies. I'm going to say it was early 2000s. So we were very, very cautious about the consequence of inducing adenovirus, giving too much adenovirus intravenously. So what we went on to look preclinically, and then we took it to the clinic, was trying to identify optimal vaccination regimens that would induce these liver memory T cells, looking for alternative routes of administration and also potentially looking for other types of vaccines that could have the same effect. And so one of the first things we did was we looked for alternative ways to give the adenoviruses. So we thought perhaps if we tried subcutaneously, this could also help. This is just demonstrating that, yes, subcutaneous did start to see antigen expression in the liver, but we needed a much, much higher dose of the adenovirus to get the same level of antigen expression. And then when we actually started to look at T cell responses, so the number of these were T cells in the liver, what we started to see that there was this strong relationship between the size of the response and the number of antigen-specific tissue resident memory cells. So it didn't really matter the route that you gave the vaccine. Really what mattered is inducing that immune response in the liver. We looked at alternative vectors. So we loved our adeno, we loved our MVA. So we did some comparative studies. We gave adeno first, adeno followed by MVA, MVA, adeno, MVA, MVA. Quite complicated. The point I want to bring is that while both combinations led to high numbers of antigen-specific T cells in the liver, when we looked at which ones were best at targeting that T cell responsively, we found that adenovirus was best. So if you looked at the number of antigen-specific cells in the liver following adenovirus given intravenously, it was much better at targeting those T cells to the liver than the MVA. But the point I want to show is that adeno MVA followed by, sorry, that's one there. Importantly, MVA given IV also induced really, really high numbers of CD8 T cells, but that's because it was inducing global immune responses. So if you gave adeno MVA, you saw really, really high numbers of circulating antigen-specific T cells, but they weren't necessarily just tied into the liver. And that's why then when we started to look at protection, we saw that both MVA given intravenously or adeno given intravenously gave us the same level of protection against malaria channel, because it goes back to the concept that protection is really dependent on the number of circulating CD8 T cells you have. Because both of these regimens induce the same number of circulating T cells, and therefore we were getting the same level of efficacy. As opposed to when we looked much later, we started to challenge these mice 12 weeks post-targeting, we started to see this difference being slightly pulled apart. So why do we see these one adenovirus being very good at targeting the immune response compared to MVA? So we started to look at not just the level of antigen expression, but also the individual cells that are expressed. And what you can see in this not great imaging is that when we give a AdHu5 vector, we can see that a lot of the hepatocytes have taken up the vaccine and started to express the antigen of interest. In this case, it's a yellow protein, fluorescent protein, TD tomato. In contrast, the MVA only gets taken up by a few cells in the liver, whereas when we look in the spleen, we see similar levels. So the greater ability of the AdHu5 to target those CD8 T cells to the liver is probably really due to the fact that you have more cells in the liver expressing antigen. So this led to look at alternative vectors. This is, again, looking at the amount of antigen expressed in the liver following AD intravenously. Same pattern that we saw beforehand. We see high level of antigen expressed and it starts to decline. This is another type of viral vector called adeno-associated virus that is also known to infect hepatocytes and lead to antigen expression. And interestingly, although we didn't see antigen expressed straight away, we started to see the level of antigen increase overall. So we knew that this viral vector also delivered antigen into the liver. And when we looked at immune responses, so adeno compared to adeno-associated virus in terms of the total number or the memory population, we can see that both were very good at targeting CD8 T cells to the liver. And that meant that both were very good in protecting mice against malaria. What does this all mean? Well, we really wanted to advance this platform, so we also did a clinical trial looking at intravenous administration of the viral vectors just as a proof of principle to start with. So translating the data that we'd seen in mice, could we target CD8 T cells to the liver in a human clinical trial, and could that improve protection against malaria? And so here we gave an intravenous administration. It was safe and well tolerated. We also went on to try and phenotype the T cell response in the liver. The way that we did this in this particular trial that was run in Oxford is we stuck a really big needle under guided ultrasound and took a small sample of cells. The important point here is that it wasn't just a peripheral blood sample that we took. We actually took a sample of liver resident cells, and we were inducing the particular populations of cells that we were interested in, and we were also inducing a particular type of memory cell. This was part of a big challenge study. We also looked at antigen-specific immune responses. The first week, we were really sending those cells off for sequencing, and the second week, we started to look for antigen-specific responses. We only have 4 volunteers. What does the data really show? Well, maybe it's showing that, again, maybe if you give adenovirus intravenously, it can target that immune response to the liver, because in this particular volunteer, it had a much, much higher antigen-specific response in the liver compared to the circulating blood, whereas these ones look a little bit similar. Unfortunately, in this challenge, we didn't see this high levels of protection that we hoped. So despite the fact that we improved efficacy overall up to 60% compared to 21%, it wasn't enough to kind of take this further. So we went there. I think they're back in the clinic now, trying alternative combinations of these particular vaccines. All right, but where am I going with this? So when we vaccinate and we give the vaccine intramuscularly, we know that it's really good at inducing overall systemic immune responses, because that vaccine goes through the draining lymph node where it activates adaptive immune responses, and we can pick up those circulating systemic immune responses in the PBMCs, or if it's a mouse, we'll take its spleen, and we can look for antigen-specific T cells, and we can look for the antibodies. And these intramuscular vaccinations, as I say, induce really good systemic immune responses, and they're really good at protecting us from severe disease. So in the context of COVID, they're really good at reducing the rates of hospitalization and protecting against severe disease. And what we've also seen that actually if we can mix up this vaccination, so if we can give an intramuscular, we can take that systemic prime vaccine and then drive those immune responses to really get better protection because we can drive that immune response to where it needs to go. So this was in the context of malaria. We also, when COVID occurred, we thought, well, actually, we know that in the context of COVID, we actually want that immune response in the respiratory tract because that's where we're exposed to the coronavirus. So maybe we can look at strategies where we can still induce that systemic immune response, but we can also target immunity into the respiratory tract. And hopefully by using mixed route vaccination, we don't compromise systemic immunity because we can induce that systemic immune response by initial intramuscular vaccination and use the same strategy that we've used for malaria, but we can target then immunity to the liver by giving that vaccine nasally. Okay, so in this case, we started to look at alternative ways that we could administer the adenovirus so that we could induce mucosal immunity. And so one of the first experiments we did was we compared intramuscular vaccination to intranasal. We also looked at oral delivery, so it went into the gastrointestinal tract, and we looked at sublingual. And then we looked at sampled a whole different variety to look at antibody responses and cellular responses, looking at systemic immunity from the serum and the spleen versus mucosal immunity by taking some of those respiratory organs. And what we found is that just what we'd seen previously, that when you give adenovirus intramuscularly, you induce really good systemic T-cell responses, but when you give that vaccine nasally, the systemic responses are slightly compromised because you're not getting as much vaccine, but you actually are able to produce a really, really good T-cell response in the lung. So you're kind of compromising your systemic immunity, but by inducing tissue-specific immunity. That's in terms of T-cell responses. And the same was also true if we looked at antibody responses. So systemically, intramuscular versus intranasal gave you almost equivalent responses. If you took a sample of the fluid from the respiratory tract, we saw better systemic immunity when we gave that vaccine intranasally. This is talking about total IgG, whereas if we look for those antibodies that are really important at blocking infections at mucosal sites, such as immunoglobulin A, what we saw that actually we only saw immunoglobulin A if we gave a vaccine intranasally. So going back to that idea of, well, do we really want to compromise systemic immunity? We started looking at mixed route vaccination. So priming the immune response with an intramuscular vaccination and then targeting that immune response through intranasal delivery. The take-home message from this is when we did that, we were able to see both systemic immunity and we were also able to see really nice population of lung-specific T cells, both CD4 T cells and CD8 T cells. So the same pattern that we'd seen beforehand. So we can prime intramuscularly and then we can drive that immune response to the lungs by giving intranasal adenovirus. And we also saw that with antibody responses. So by priming intramuscularly, we could really drive mucosal immune responses by giving a second vaccine, which is a nasal delivery, and really inducing the type of response in the respiratory tract that we know leads to better protection. So IgA that we can pick up in the bronchial lavage fluid, and these antibodies also acting effectively. Okay, but there's one problem, well, not one problem. One thing we did notice though is that in these previous experiments, we'd given a pretty large volume of vaccine intranasally. So we gave the mice about 25 microliters, and we suspected that that wasn't just locked to the nasal cavity, it also would go deep into the lungs. So we thought, well, where do we need that vaccine to be? Does it need to be in the lungs, or can we induce enough immunity by just keeping that vaccine up into the upper respiratory tract? And what we see is that the that inducing that mucosal response, so inducing T cells in the lung, particularly tissue-resident memory cells, was really actually dependent on there being enough vaccine present so that it would actually go deep within the lung. So you really needed to give a high dose of vaccine to get it into the respiratory tract to induce that mucosal immune response. The other way that we saw that is if you, so the way that we vaccinate these mice is we sedate them with anesthesia so that they'll actually inhale all of that. If you don't anesthetize the mice, actually what happens is that they just spurt it straight back out into your face. So we need these mice to be anesthetized so that they can take all of that volume deep within the lungs. And so why are we seeing this very, very big difference in immune responses? If we just keep it up into the nose by giving a really tiny volume, we don't start to see that mucosal immune response. Whereas if you give it to the entire respiratory tract, we actually are now starting to see better responses. What is driving this anatomical difference? And it leads us to this question about what is actually happening. We kind of talk about the respiratory tract as being one entity, but actually we know that it's very, very different at different parts within the respiratory tract. So we break the respiratory tract down to the upper respiratory tract that would really encapsulate your nose and just the top hand part. And if you think about what's happening up in your nose, it's all about filtering out environmental proteins and kind of protecting you against things, but at the same time, you know, filtering things out. So there's lots of mechanisms in place there to kind of clear out the immune response. In contrast, when you get deep within the lungs, the function of the cells deep within the lung also changes. So now you're turning down, or you've got really alveolar cells that are really about gas exchange. And there's less exposure to pathogens down here, meaning that if a pathogen does get down here, it's pretty serious and pretty dangerous, and you really need to respond to it. We know that epithelial cells are the first point of contact with these, with a virus that really initiates that immune response. So knowing that the cell composition is very, very different between the nasal cavity and the lower respiratory tract, but also knowing that there's overall functional differences, we wondered whether there is perhaps a difference in the way that epithelial cells from these different sites initiate immune responses. So could it be that epithelial cells up in the nasal cavity actually just are not very good at inducing immune responses, whereas those epithelials deep within the lungs actually are inducing better immune responses? And that's why we see the difference, that if we only give enough vaccine to stay up in the nasal cavity, we don't see that really good mucosal response, whereas if we're able to get vaccine deep within the lungs, now we can start to see that mucosal response. And that's a pretty challenging thing to do in mice because we've got mucus, we've got airflow. So one way we started to look at this potential difference in epithelial cells, if we were going to look at an organoid culture system. And this is some work of a PhD student I have. So what she does is she takes epithelial cells and she generates these 3D cultures. So what you can see here is these are cells from the nasal passage. They develop these 3D structures. They've got different stratified epithelial. I don't have the picture there, but these form different types of organoids that are that have been documented in the liver. So you have these called solid organoids. We would call these cystic organoids. She's also able to generate organoids from lung-derived epithelial cells that have these different characteristic structures. So we think these are coming from alveolar cells. We get solid organoids, we get cystic organoids. So you can generate these nasal-derived epithelial cells versus lung-derived epithelial cells in these 3D culture systems. And now we can start to actually compare the epithelial cell response without the complications of in vivo challenges. So being able to deliver a vaccine to specific sites, we don't have to worry about mucus. We can, start to actually look at the role of epithelial cells. And there's one type challenge that we currently have, is that the way that organoids grow is they grow so that where you would normally administer the vaccine is what's called on the inside. So they grow in around circular structure, and so within the circular structure is actually what would typically be the lumen of the epithelial tract. So if you're trying to test vaccine responses, either what you have to do is you have to stick a needle into there to try and see if there's vaccine uptake, And that's not very good for kind of trying to do lots of screening. So she came up with a particular protocol where she could essentially flip these organoids. So by culturing these cells, she could flip them so that the apical surface is on the outside. And now we can start to kind of compare vaccine uptake by these different types of epithelial cells. So nasal epithelial cells versus lung epithelial cells. And to do that, we used an adenovirus expressing a green fluorescent protein. And the first thing we saw was that actually, despite our thoughts that actually maybe nasal epithelial cells can't be infected by adenovirus, both nasal and lung-derived epithelial cells can take up the vaccine and start to express the protein, because you see green fluorescence here. Importantly, we don't see it with a virus that's not expressing GFP. But the important thing that we see as well is when we do this 3D imaging, what's interesting to see is it's not all cells that have taken up the vaccine, it's just a very few select cells. And so what we're looking at now is trying to identify, well, what are the cellular targets of these particular adenoviruses? Because if we're going to develop a mucosal vaccine, knowing that adenoviruses is actually really good at inducing mucosal responses, maybe we can look at ways that we can really specifically target to individual cells within the respiratory tract. So we've done this in mice. We're also looking at ways that we can do this in humans, because what we would like to set up now is we want to set up a screening platform so we can test other types of vaccines. I'm conscious of the time. What I was going to say is what we're also doing is we're looking at not just the adaptive immune response, we're trying to understand what are those early changes in immune responses that are driving these differences in vaccines that I talked about before. So we know that different types of vaccines and different ways that we administer the vaccine can really change the adaptive immune response. What is that coming from? So this is just to say that I've got a honours student who's been looking at the role of different types of antigen presenting cells and looking to see whether there's a difference in the cells that present antigens when you deliver vaccine to different places. So if you deliver it into the nasal cavity, what type of cells are expressing GFP compared to the lung? We've also looked in muscles. So is there a difference between the cells that take up and present the antigen when you give it in the muscle versus the draining lymph node? So in summary, hopefully you'll agree that what we've been able to see from studying all of these different types of vaccines, that the immune response, the adaptive immune response that is induced is really driven by the type of vaccine that you use. So if you're really looking to drive cell-mediated immunity, perhaps what you really want to be using is a viral vectored vaccines. If you really focused on a disease where you need a lot of antibodies, then perhaps a protein or a virus-like particle in adjuvant will be good. We know mRNA vaccines are also very good at inducing antibodies, and adenoviruses are not bad. They can still induce it. If you're perhaps looking for something where you don't know what you need, so maybe you need T cells plus you need antibodies, then maybe you can do a heterologous vaccination because it gives you the best of both worlds. I haven't quite explained it very well, but what I wanted to highlight is that an intramuscular vaccination is great in inducing systemic immunity. What we've discovered, but by mixing up the routes of vaccination, we're able to target immune responses to specific sites. So in the context of malaria, we can induce a systemic immune response and then target those T cells to the liver to give us better protection against malaria. And in the context of respiratory infections, we can induce a systemic immune response, and then we can also target those T cells and antibodies into the respiratory tract by delivering adenoviruses. What my group is really focused on at the moment is trying to understand and compare immune responses that we see from different vaccines. And so we're looking at both those adaptive immune responses, but also what is going on early in that immune response. So how do changes in antigen expression shape the adaptive immune response? Because our real goal is to understand better if we need to induce a particular type of immune response, what cells do we need to target, or how do we need to deliver that vaccine so we can target the type of immune response we induce, but also the site that how can we ensure that that is induced in the right site? And so, as I said, what we're really focused on is understanding what are those differences that arise early. So what are those differences in innate immunity that drive changes in the adaptive immune response that could be dependent on the particular, that leads to being able to better select the type of vaccine. Because if another when the next outbreak comes along, if we know the type of immune response we're able to do, we'll actually be able to better select the types of vaccines by knowing which types of vaccines induce these particular types of immune responses in terms of the phenotype and their function. So it also means that we can look for more effective vaccines for targets that are already for diseases that are already out there. So if we're really trying to improve influenza vaccines, that maybe we can select better vaccines or better delivery modes so that we can actually induce antibodies and T cells in the lung that hope that we actually leads to better protection. Or if we're looking for a therapeutic vaccine against hepatitis B and we're trying to induce T cells, well, maybe we can come up with a combination of vaccines in a delivery mode that actually will lead to effector T cells in the liver that can clear the infection. And with that, it just leaves me to thank a lot of people that are involved in the work. I'm sorry, I've covered loads of topics, but there were some really key people when I was in Oxford involved in the liver stage malaria work. So I showed you data from Anita and Benedict, but also lots of PhD students and postdocs that really contributed to this work in the context of liver stage malaria, but also COVID. There was a lot of work done in COVID, which has yet to be published, but building on that same concepts. And now in Australia, I've got some really great PhD students and honours students that have really kind of laid that foundation now that we're starting to look at nutting out what are those early changes that can really drive adaptive immune response. And with that, I just thank the funders that have come from different sources, and also thank you very much all for listening, and happy to take questions.
Yes, thank you for such a wonderful presentation. It's good for research and development of vaccines. Very nice. I request the audience to post their questions on the chat or raise their hand using the available icon so that the presenter can take the questions.
I'm just going to stop sharing so I can see properly on my screen.
Yeah, it's fine. Sure.
And I'll try and open the chat.
Yes. And post the questions on the chat. So you can raise your hand so that
If there's no question, that's okay. I'll also happy to answer questions on emails if anyone thinks about something they don't have it. Oh, we've got a question.
Yeah, from Rinda. Yes, Rinda Javiero, kindly go ahead. Yes, Rinda. Yes, Rinda, kindly go ahead. Rinda.
Hello.
Hello.
Hi.
Yes, we can hear you.
Hello, good afternoon.
Yes, good afternoon.
Thank you so much for the opportunity. I'm really glad to be part of this presentation. It was amazing. I'm also in the field of immunology. My question is not directly related to tuberculosis or malaria. It's rather general. It might be a bit rounded, but I hope I'll be able to deliver the question. So the point is in terms of the pathogen, the specific pathogen targeted by a vaccine, and the route of administration, as you explained in the earlier slides. Would you kindly clarify the logic behind having a humoral-based vaccine for a viral infection, case in point, HPV vaccine, because viruses are intracellular. We know that the infection, the pathogenesis is within the epithelium layer, but then the HPV vaccine targets antibody production.
Yep.
So,
how does it work against a virus?
yes, kindly.
Yeah.
Because
so if you think about a virus infection, before it becomes intracellular, it's extracellular. And the way that viruses infect is they need to have a receptor and they'll have a target on the cell. So the way that that antibody is working to start with is it's stopping that virus from actually infecting that cell. It's the same with SARS-CoV-2. That's how the vaccine works, is that if you make enough antibodies, you can bind and neutralize that virus before it's able to infect the cells. And I guess antibodies are then also still important because that virus infects that cell, but it also generates more copies of it. And then those viruses are going to get out and infect other cells. So it's not just the first time that you, the first infection that you want to stop the overall infection and spreading of that virus. You also want to stop it from being able to infect other cells. So that's how the antibodies work in that context. They're interfering with the ability of that virus to infect and that virus to infect more cells, if that makes sense.
Okay, then would it be right to say that they target the pre-cellular infection stage, if that makes sense?
Yeah, so it's stopping it when that virus is extracellular because it doesn't spend it while it's replicating, it's intracellular, but there's also a stage in its life cycle where it's outside of that cell when it's infecting other cells and when you first receive the virus. So that's how the antibodies are working, is they're stopping it when it's outside of the cell.
Okay, very good. Thank you.
No problem.
Yes, there is another question from Ignatius Mungangi. He says that given the logistical challenges in low resource settings, which administration routes hold the most promise for balancing immunogenicity with practicality?
Yeah, that's a great question, and that's really something to consider. I guess that's why I very cheekily pointed out the fact that while irradiated sporozoites are highly highly protective, maybe they're not going to make the best strategy because doing intravenous injection of cryopreserved sporozoites might be the best. When we think about, so my focus is shifted a little bit to respiratory viruses. I think that perhaps mucosal delivery is not, if we get over working out where we need to deliver it, mucosal delivery actually could be not such a challenge to administer, because if you can just squirt it up the nose, maybe that actually is a really effective way of getting the vaccine out. I think that's what you're getting to. We've got to think of practical ways of giving these vaccines. If intramuscular vaccination gives us enough protection, then actually probably we stick with intramuscular vaccination. It comes down again to cost benefit. You know, we need something that gets to people who need the vaccine, and we need it to induce enough immune response to be protective. But we definitely have to think about how we're going to deliver that vaccine, and making sure that the way that we give the vaccine is appropriate, like it can actually be delivered. I think that's where we're getting to with that question. I hope that's okay, that answer.
Yes, there is another one from Ikecho. Thank you for the presentation. I could love if we could talk a bit more about strategies towards achieving greater efficacy, these factors.
Yeah.
Yes.
So I guess it kind of comes up to balancing those things. So how do you get, what is what's the word I'm looking for? I want to say effective, but how do we get better vaccines? So, we spend a lot of time in Oxford looking at ways that we can improve the overall immune response. So can we deliver our vaccines with adjuvants that lead to higher levels of immune responses? That's certainly what happened in the context of the R21 vaccine. So trying to find the best adjuvant to go with a virus-like particle. Maybe we can we can manipulate the viral vectors so that actually they target only the cells that we know are good at inducing that particular type of immune response. That's a bit complicated because we also have to understand the biology behind that, which we really don't understand. So once we have a better understanding of the immune system, maybe then we can design these better vaccines. What I would say is in the vaccinology field, everyone tries everything. So everyone's always looking for ways that it can improve the immune response, that it can improve efficacy. It's always on the tip, it's always trying to find the better vaccine in that context.
Yes, sure. There is also another question from Lilian. Thank you for the nice presentation. My question is, you meant that you're looking for ways to administer the antigen to humans. As this started, and briefly, do you plan to achieve this? Then there's another one is, so far we have the LTSS vaccine trials. What difference are you trying to implement to ensure that vaccines avoid mutations or changes that occur in malaria to hinder vaccine development? Can be wide. Yes.
So I'm going to answer the first part and then I'll come back to the second question. So if we're talking about the work we were giving adenoviruses intravenously, so that study was done in humans, in malaria naive individuals in Oxford. So that started, it showed pretty good efficacy. Again, doesn't really seem like the highest deployable vaccine because it's intravenous administration of viral vectors, potentially not the greatest thing to do. When I left Oxford, they were looking at different combinations of things. So it was still intravenous injection. Again, we need to look at other ways to do that. Perhaps we look at where would we go? We need to look at vaccines that actually you can administer by intramuscular injection that would give that same level of response. So that's kind of halted the liver stage malaria vaccine. At the same time, being more of a shift towards an antibody-inducing antisporozoite vaccine. The liver stage is a challenge. I don't know where we're going to end up with that one. In terms of so the second part of the questions, so we have RTSS vaccine and R21. So at the moment, both those vaccines express exactly the same antigen. So they're really not addressing the potential of the sporozoite to mutate. I guess the way that people are thinking about that is they're sequencing CSP protein from a huge variety of different isolates to checking to see, how many changes are occurring. I guess, if you can block the infection, then the parasite can't slip through. The way that people look at that is they would make second generation vaccines with new antigens depending on the mutations that occur. If that kind of answers that question.
Yes. There is also another one closely related. There is on the Q&A, Bumura Mashozi from South Africa. Great talk, RX. I had a question: if one vaccine procs infection at the liver stage and another targets merocytes in the blood, could administering them in sequence enhance overall protection? Then when you mentioned heterologous vaccination, my follow-up is, there is currently heterologous, is there currently a heterologous malaria vaccine available? If they are available, is there an improved protection? Yes.
So I'm going to go to the first part of that question. So one of the programs in Oxford is actually looking at ways that you can have combine blood stage vaccine with a liver stage vaccine and a transmission blocking vaccine, because really I think that's the best way that we're going to actually lead to eradication of malaria, by making sure we protect in each stage. So there's definitely a program in Oxford looking at that. So how do we combine blood stage vaccine with a liver stage vaccine, and does that lead to better protection? I'm going to ask you to repeat the second half of the question, the second part of that question, if that's all right, Clive, because I don't think I can see the Q. Oh, hang on, I see the Q&A.
Yes, let me repeat the second half. Then when you mentioned heterologous vaccination, my follow-up is there currently heterologous malaria vaccine available? If they are available, is there an improved protection?
With a what type of malaria vaccine?
She's asking about heterologous malaria vaccine.
Oh, heterologous. So it's not licensed. So heterologous being one viral vector plus another viral vector. It has, is that right?
Yeah.
Yeah. It got as far as phase 2 trials in Khalifi. There was a study in Khalifi, and there was also a study in the Gambia. It still didn't give the greatest level of protection, so it's kind of gone back to the drawing board to look for other ways to induce that liver stage immunity. At the same time that we were still looking at heterologous vaccines to induce liver stage immunity, there was that other program which was about antisporozoite vaccines, which really is the R21 and the Oxford malaria vaccine. So that's kind of overtaken the work that we're doing in liver stage malaria.
Yeah, sure. Again, we'll wait for 1 or 2 minutes for another question on the chat. Alex, maybe you could give your closing remarks. There is more one question from Regan. She says that I have another question. I'm wondering that today there are so many vaccines. There are any studies so far that have assessed effects of all three vaccines injected to kids or adults, specifically injected to the same person in 3 months, like kids?
Yeah, so I haven't been part of this, but I know that there's definitely been studies looking at what is the impact if you're giving multiple vaccines at the same time. Do you get competition between those immune responses? I think what's been seen, and that's why you have the licensing and the EPA programs, is that administering multiple vaccines doesn't appear to impact the immune response against those individual components. But that's certainly something that does get checked. So a lot of, before a vaccine is licensed and introduced into the EPA scheme, they make sure that, the addition of an extra vaccine isn't compromising immunity against any of the single components.
Sure. There is another one from Linda. With the heterologous vaccine trials, was there an impact of the adjuvants of choice in the observed responses?
So the heterologous vaccination with viral vectors, we haven't used adjuvants in that. It's just the straight virus, the viral vector, because the viral vector alone induces enough an immune response. As far as I'm aware, there haven't been what you would call a heterologous vaccination with R21. So when you have R21 with one type of adjuvant and then trying a different type of adjuvant, those studies haven't been done. Certainly not in Oxford, as far as I'm aware, and it certainly hasn't happened in humans. So I can't actually answer that question.
Okay. We'll close there because we are at 12:30. Maybe Alex, you will give your closing remarks. Really appreciate.
Well, I just have to say that vaccinology is an amazing field. I got into it because I just was amazed by the power of vaccines. I think it's one of the greatest things that we can do to level the playing field in society. So it's really encouraging that so many people are interested in vaccinology. And, if anyone's in Australia or they're coming this way, please get in contact with me. Always there to provide opportunities for people if I can. But stay in vaccinology. It's an amazing field. And thank you very much for letting me talk to you today.
Yes, thank you very much, Alex, and thank you very much to everyone in this webinar. We would like to hear about your experiences and what else you could like us to discuss in upcoming forums. Before we close, kindly requesting that you take a few minutes to respond to the post-webinar survey posted in the chat. There is a link there for that. Have a nice afternoon.